Good morning, everyone. So welcome to the day 2 of HEAD Summit, and we are going to start with a very exciting session on clinical informatics. And health informatics isn't complete without clinical informatics. So we are going to hear about the translational aspects of health informatics as they can be applied to health in clinics and on the bedside. We have a stellar lineup of speakers, and it's my pleasure and honor to introduce the first speaker. I'll be co-chairing this session along with Dr. Gaurav Ahuja from IIIT Delhi, and it cannot be better than anyone else than Dr. Vinod Scaria, who is a clinician turned computational biologist, and he's the first speaker of our day. Dr. Vinod Scaria is a principal scientist at CSIR IGIB, Institute of Genomics and Integrative Biology, and is also an adjunct faculty at IIIT Delhi. Dr. Scaria is involved in creating novel methods and resources for analysis and annotation of genomes and understanding of the functional impact of genomic variations. He has led and been a part of numerous large-scale genomics projects, including the Asian Genome Diversity Project, and he's a member of the Hugo Pan-Asian Population Genomics Initiative Task Force, and also the co-founder of the Genomics for Understanding Rare Disease in the India Alliance Network. Dr. Vinod Scaria did his undergraduate medical education from Calicut Medical University and his PhD in computational biology from University of Pune. He has more than 90 publications in international peer-reviewed journals, a book and 2 book chapters to his credit. And he's also on the editorial board of numerous journals, including PLOS ONE, PSJ, Journal of Translational Medicine, Journal of Orthopedics, and International Journal of Rheumatic Disease. He's a recipient of the CSIR Young Scientist Award for Biological Sciences in 2012 and a Kavli Frontier of Science Fellow of the US National Academy of Sciences, 2015. But today, I've requested Dr. Scaria to speak about his COVID work, which he has done some extensive and stellar work in the last 1.5 years on unraveling the COVID spread. So over to you, Dr. Scaria. We look forward to your talk.
Thank you very much, Stav. I hope my slides are visible.
Yes, they're visible.
So thank you, and good morning to all of you. It's always difficult to be the opening batsman early in the morning. But over the next 30 minutes or so, let me briefly introduce what we have actually learned from looking at the SARS-CoV-2 genomes from India. Now, before I start, it's also customary to introduce the topic and also give a brief history about the field in interest of the students out here. Now, microbiology, as we know today, is heavily indebted to this one man, Anthony von Leeuwenhoek. 400 years ago, he discovered what we today know as the microscope. And practically all of microbiology that we know today is thanks to this instrument, which was a handheld device to start with, and using which he had a fairly large collection of drawings from the world around him, which are not visible to the naked eye, but now are visible through this instrument. And incidentally, by profession, he was a merchant and a municipal official, and by passion, a scientist. Now, while 400 years of microbiology has been revolving around this one single instrument, it's only a decade or so ago that there has been a paradigm shift in looking at microbes using a very different kind of an approach. And it is sort of summarized in here because the earlier approach of looking at microbes out of epidemics or out of infection outbreaks involved a very laborious process of culturing and isolating the virus or the pathogen, and then observing them under the microscope or under a specific type of microscope, which can look at very small pathogens. But around a decade ago, this underwent a really big paradigm shift. And the concept or the underlying basis of this paradigm shift is that practically all living organisms to have the genome, whether it's a DNA genome or an RNA genome. They do have a genome. And if you can sequence the genome of the organism, then you could pretty much identify the organism without having to go through the time-consuming and the laborious steps. And this is sort of exemplified also in the current pandemic. As you see here, the picture that I have here is essentially the steps from the paper which described the Wuhan U1 genome, which was the first genome to be available in public domain from the Wuhan pandemic. Now, what this paper describes is that the sample was taken, no culture isolation was done, was directly taken up for RNA isolation, RNA and DNA isolation, and sequencing. And sequencing identified the pathogen, not just identified the pathogen, but also revealed the whole genome of the virus. So in other words, it is not just detection, but also genomic characterization at the same time. And unlike all the other outbreaks that has happened in the past, the identification of the index patients and the genomic characterization took days, if not weeks, and not months and years as it used to happen before. And this increasingly is going to happen in the future, as you see here on the right side of the panel, as the cost of sequencing genomes continue to dwindle quite rapidly. Now, why do we need to sequence genomes, especially of pathogens like of SARS-CoV-2? Isn't just one genome enough and available? Why should we really sequence genomes over and over again? The answer to that is because of the evolution of the pathogen. Every organisms evolve, includes humans. The only difference is that the rate of evolution of an organism differs very distinctly different from each other. In the case of a virus like SARS-CoV-2 with a modest substitution rate, the virus mutates approximately 1.48 in 10 to the power minus 3 times per base per year. That roughly or approximately translates to approximately 3.7 mutations a month. Now, why is it important? It is important because as the virus transmits from individual to individual, and as the virus gets more and more people infected, it accumulates additional genetic mutations, and as they accumulate genetic mutations, they leave what we call as fingerprint of the virus behind. Now, this fingerprint of the virus is what is really important. And as you see here, the chain of events, the virus mutates or accumulates additional mutations on a pre-existing background of mutations already present at that point in time. So therefore, if you manage to isolate the virus at different time points or time intervals, you could understand how did the virus mutate, where did the virus come from, and how did it spread. Now, assuming a constant rate of mutations across time, you would assume that in a real life situation, all the kinds of virus mutations, in this particular case, for example, 123, 124, 1235, and 1246, would be equal in proportions or near equal in proportions. But that's not really what you observe, because at points in time, you need to have, or you do see viruses which are much larger in proportion compared to the other viruses. And this is what we call as emergence events. And emergence events essentially means that you have a disproportionately large number of a particular kind of a variant or a mutation compared to the other viruses in circulation. Now, emergent events are not just because of viral mutations and virus advantage. That is a very simple way of looking at it, but it could also be an artifact of human events, like, for example, super spreader events or congregations, which would take the exactly same virus to a large number of people harboring the same virus mutational fingerprint. Now, this is what we look at when we do whole genomes. And the idea of looking at whole genomes is to also understand and give an insight about these emergence events that happen from time to time. Now, until mid last year, there was not enough tools to sequence genomes at large numbers. Yes, you could sequence genomes in dozens, in singles or in dozens, at the max in ranges of 100, but it was not possible to sequence very large numbers of viruses until then. And one of the first FDA approved tools to be able to sequence genomes at very large number is this approach which is known as a COVID-seq, which was brought together by Illumina, which relies on the principle that you could attach barcodes in very large numbers, 384 to be exact, in a lane, and therefore you could multiplex a large number of viruses for, for sequencing. And one of the first real life applications or rather studies, clinical studies in this particular area happened in IGIB in India globally, in the lab of Dr. Sridhar Subbu, and of course contributed by a bunch of young researchers out here. And the idea of this approach and this validation was now you could sequence anything from 750 to around 3,000 samples of the virus in one single go at a quite affordable and a quite short timeline that you could produce not just reports in terms of whether they're detected or not, but also in terms of understanding the genetic epidemiology of the virus in terms of what variants are there, how are they emerging, so on and so forth. Now, this has been a significant landmark because this enabled genomic surveillance to happen in very large numbers. As I suggested here, it could sequence anything modular from 750 to 3,000 samples. Now, to do such genetic epidemiology, of course, you need to work with the states because health in India is a state subject. And therefore, we collaborated quite extensively in the mid part of the last year with 7 states across the country to sequence a large number of genomes. And this program was pilot funded by CSR in terms of understanding the genetic epidemiology and also in an attempt to pilot the COVID-seq approach for genetic epidemiology of SARS-CoV-2. And what we learned from the early part of the pandemic is that compared to the 6, or rather, India largely comprised of 7 lineages, and out of the 6 was largely prevalent in other parts of the world. And there was one distinct lineage that you see here in purple, which was found in quite significant numbers in India, but not much in many other parts of the world. And since this was a significantly large cluster in India, we sort of named it IA3I, I to signify that it is a significantly Indian cluster, and A3I to suggest that it was an outgroup or a divergent group from the A3 cluster of SARS-CoV-2. The A3 cluster is what you see here in green, and this essentially are the travelers from Iran who came into India. But nevertheless, the A3 cluster is not much related to the A3I cluster because as you see here, there was a much earlier divergence event from the green dots that you see here. And then therefore, this was a cluster which diverged much before the A3 cluster appeared in India. Now, this cluster, we could follow up using the genomic surveillance data generated by us as well as by other colleagues across the country. And what we found was quite remarkable in the sense that the proportions of the A3I cluster was significantly different in different parts of the country. It had very large proportions, for example, in Delhi, in Andhra Pradesh, in Karnataka, and so on and so forth. And but of course, it had much lower or almost nil frequencies in places like Kerala. Now, this was very perplexing because this was a very recent origin of the virus or the virus lineage. And by using computational tools, you could now sort of look at the genetic epidemiology or trace back the time of origin. And as you see here from this cluster diagram, we sort of suggest that almost all these individuals or the viruses that were isolated at different points in time from different parts of the country all seem to have diverged from one single event, or what we call as a super spreader event, which took this virus far and wide to different parts of the country. And of course, over time, we did have an opportunity to trace this virus across timelines. And as you see here, the largest numbers was in and around March 2022, March 2020, but was later taken over by this blue colored lineage, which is a mutation containing lineage D614G. Now, this is what you could understand as an example from looking at SARS-CoV-2 genomes in large numbers, if properly represented from different parts of the country. We were also fortunate to have looked at the data in a very different perspective into looking at outbreaks that has happened in similar fashion across the country. Now, just to mention that this is not just one outbreak that happened, there are multiple different outbreaks of different proportions that has happened in different parts of the country in early part of the pandemic. And many of these outbreaks came from the southern states, from Andhra Pradesh, Karnataka, Tamil Nadu, Telangana, so on and so forth. And this was an opportunity to understand that the early part of the spread in 2020 was largely contributed by local and large outbreaks that has happened. And many of these outbreaks could be epidemiologically traced to marriage events, to super spreader events, to markets, to hospitals, and so on and so forth. And one such example is the outbreaks in Kerala. Kerala is a southern state which has a quite significant expatriate population which returned from different parts of the world. And they did have to go through a quite stringent quarantine procedure. Now, it was expected while looking at the data in Kerala that the lineages would have quite significant representation of international lineages. But what we found was quite remarkable and significant in the sense that almost all the outbreaks in Kerala were linked to just 3 events or 3 clusters, what we call as K1, K2, and K3. And none of these 3 clusters had an international event or international compatriate. Now, what this would mean is that, and K1, K2, K3, as you see here, almost always came from the nearby states. And what this really suggests is that the early part of the spread in Kerala was largely contributed by interstate travelers rather than international travelers. And many of these interstate travelers could be traced to the nearby states, like, for example, from Karnataka or from Orissa and so on and so forth. And this was really significant because this could also suggest that the policy of quarantining people for international travel or who have returned from international travel was quite remarkable and quite successful, that it didn't contribute much to the local spread of the virus. But it could also suggest that the quarantine of individuals who traveled through the borders across the states was probably quite lax and contributed to the outbreaks locally within the state and outside the state. And therefore, this is to also suggest that genomic surveillance can generate evidence, not just generate evidence, can also validate a policy in terms of policies like public health policies like quarantine, the time and duration of quarantine, and also suggest positive measures in terms of how to curtail the spread of the virus. For example, here, quarantining interstate travelers. Now, taking from there, Kerala has been one of the first states to actually initiate a very systematic program for genomic surveillance in November 2020. And this was a very, what I call, well thought through and elaborate public health measure, which involved 12 government medical colleges, the state and regional public health laboratories, 2 university centers, the Inter-University Center for Biomedical Research and the Central University of Kerala, and 14 district surveillance units, 1 district surveillance unit in every district, which could now act upon the evidence. And the idea was to do a very systematic survey, collecting 100 samples from each district every month, and be able to understand what really happened in the district in that particular month. And over time, this sort of surveillance program has been very successful in identifying new lineages of the virus, has been very successful in informing public policy, like, for example, the public policy of double masking, where Kerala was one of the first states in the country to initiate a policy of double masking, in the view of the fact that alpha was emerging quite significantly in the state along with delta early in April. And as delta was increasing quite significantly, there was early lockdown and augmentation of the beds and capacity for oxygen. And this all contributed to the successful containment of the spread of the pandemic in the state. And the outcomes of this is quite evident in the fact that the per capita mortality due to COVID has been one of the lowest in the state. And this is also exemplified in the much lower excess deaths in the state of Kerala compared to other states in India and even other countries abroad. Now, genomics can also be used to understand basic biology and also understand the genetic underpinnings of very curious clinical observations, like, for example, persistent infections, reinfections, reactivations, and of course, much recently with the vaccines, breakthrough infections. I'll give a few examples of how genomics could be used to understand such very basic clinical observations and differentiate them. And the first of these cases came from a hospital very close by in Noida called the Government Institute of Medical Sciences, which had a very systematic program for genomic surveillance of SARS-CoV-2. And in fact, the Government Institute of Medical Sciences has been one of the first collaborators in genomic surveillance with IGIB. And this hospital had put all their healthcare workers on a very systematic RT-PCR surveillance early part of the pandemic. So as part of this surveillance, we did identify 2 individuals who turned out positive at 2 distinct intervals. Both of them are healthcare workers, and they were asymptomatic. In other words, they didn't have any major symptoms that necessitated that they understood that they had an infection. And at 2 episodes, both these individuals were asymptomatic. Now, we could do the genome sequencing for these individuals at these 2 intervals of the virus, and what we show here is E1 and E2, which is episode and episode 2 for I1 and I2 for these 2 individuals, individual 1 and individual 2. And what you could see from the genomic sequence is that the viruses do have a very distinct mutational profile. In other words, these are 2 distinctly different viruses, which sort of confirms the fact that this is a case of reinfection. And therefore, we could contribute to the global literature one of the first global cases of asymptomatic reinfections in healthcare workers, also suggesting that healthcare workers should possibly be put on RT-PCR or molecular surveillance mechanism rather than by looking at symptomatic incidence of the disease. Now, this is another similar clinical observation where you have individuals who test positive on 2 different occasions. In this particular case, this is a child from Medanda who was on chemotherapy, who tested positive on 2 different episodes. We could isolate the virus from these 2 episodes, E1 and E2. And what here you suggest is that both the viruses in E1 and E2 do have exact same mutational profile. But in other words, this is a case of reactivation of the virus. The first and the second episode of the infection did have the exact same virus, but the individual also did have an intermediate period of symptom or RT-PCR free. That necessarily means that the virus had actually gone back into some kind of a remission in this individual. Now, of course, there are many more cases of asymptomatic and symptomatic reactivation of the virus now reported in literature, but this remains one of the first such cases of reactivation, which was genomically confirmed. Now, as vaccines have been implemented in the country from early part of this year, at least starting with healthcare workers, so genomic surveillance also provided very useful insights into reinfections after vaccines, that's what we call as breakthrough infections. And this came from Kerala and 6 healthcare workers, which suggested that early breakthrough infections were largely contributed by the variants of concern. And in global literature, this is the second report of breakthrough infections confirmed by genomic surveillance. And of course, beyond the state of Kerala, now we have multiple different states which have surveillance programs, including the state of Maharashtra, and also INSACOG, which sort of coordinates the national program for all the states. And today we have crossed 50,000 of genomes from all the states. And we also were fortunate to have maintained the database or the dashboard for the entire data set. And to make data access much simpler and easier, today now we have implemented the APIs and the beacons, which would allow for anonymous access to the granular data of mutations from this data set. And this is essentially a technological advancement over the current ways of data sharing. Like, for example, if you go to GSAID, you could not access in a much granular fashion, or in many ways, you cannot access the dataset programmatically. Now, using the Beacon API, we can now make this data available programmatically. And in fact, a large number of individuals are accessing this datasets programmatically and could also incorporate the datasets and knowledge into their analysis workflows without having to download and re-download and reformat the data for use every time that they want to use. Now, such genomic datasets can also be useful to inform public policy in terms of, for example, RT-PCR primers. And as you know, that RT-PCR primers are based on the fact that you have primers or probes which could bind to very specific regions in the genome, and genetic mutations in such genomic regions could adversely affect the efficacy of RT-PCR primers. So in other words, you could look at RT-PCR primers and the sites where they could bind the genome and essentially look for frequencies of genetic variations in these sites to get a rough information about what are the RT-PCR primers which are unlikely to work in a specific condition. And this was one of the early papers in 2020, where we put together data for global sequences and sort of put together a set of RT-PCR primers, which are very unlikely to work given the current mutational spectrum, which is prevalent across different parts of the world. We do have now very specific maps like this for India, which a number of companies are utilizing to actually not just design new RT-PCR probes, but also evaluate whether their RT-PCR probes designed a year and a half ago are still efficacious as before. Now, of course, going from here, the obvious next question is what's going to really happen in the future? And if you look at the trajectory how the virus has been evolving, it largely has traversed the landscape of immune escape or largely antibody escape and of increased transmissibility. Unfortunately, each and every one of these genetic lineages that has happened in the past have happened through very systematic and step-by-step evolution process. It has never jumped across the space to become a very highly transmissible or a very highly immune escape variant. And today we have Delta, which is pretty much the predominant lineage across India and across the world. And what we are essentially looking forward is the next step in evolution. What is the virus trying to do? Is it trying to increase the transmissibility, or is it trying to increase the immune escape? And therefore, to look at such escape events or transmissibility events, we need to look at the genomic evidence of both of these factors. And therefore, we do have two preeminent databases or resources in our lab, which was curated over the last year and a half, and probably the only resources of its kind in the world, which looks at very specific evidence of mutations which cause immune escape and mutations which cause increased transmissibility. And unfortunately, I think we have sort of got a hold of the Achilles heel of the virus, because practically all of immune escape mutations are very focused into a small region in the spike protein. As you see here, this colored out here. And this also overlaps with this blue colored region, which is the receptor binding domain of the protein. Or in other words, the virus doesn't have an invisible character or invincible character rather, with the fact that additional mutations need to optimize between its efficacy to latch onto the human receptor and therefore the transmissibility, and also evade antibodies through mutations which can cause immune escape. Or in other words, there are only a finite set of mutations the virus can do to traverse the space of optimal transmissibility with optimal immune escape. And of course, such databases and tools are very useful in terms of evidence to look at emerging new viruses across the world. Now, obviously, the next question is, can we find such variants in their tracks even before they create the next wave? Looking at history, what you can say is this is possible. And this is possible, of course, with some conditions in place. What you see here is essentially the first and the second waves in Maharashtra. And what you see here in the arrow mark here is the time of origin of P1.617.2, which is otherwise known as delta. And as you see here, delta originated in time pretty much during the first wave of the pandemic in Maharashtra. It stayed low for quite some time, created the first outbreaks in Amravati and Yavatmal, and then created the wave as we know, what we call as the second wave. Now, as you see here in this graph, there was a considerable amount of time, which I would not say lost, but was not probably properly used for lack of evidence, that if you had a very systematic surveillance during the trough period of the outbreak, you could have identified the emerging viruses much early in time to institute public health measures much early in time. And that's what I'm trying to suggest, is to say that even at the max, we should be able to identify outbreaks that would happen in a local region and be able to understand what is happening and what is emerging from these outbreaks. And of course, the ideal solution would be to extend the surveillance during the trough of the epidemic, where there are no cases. That's pretty much where you should be the strongest surveillance. Now, doing surveillance when the cases are high is not really going to mean anything, as you see here. During the peak of the pandemic, more sequencing is not going to look at more diversity of the virus, but essentially is going to look at more and more of just one type of virus, which seemed to be successful. As you see here, 617.2 is the blue one out here, 6.7.1 is the orange one out here. And essentially, if you did sequence in the pandemic, you're essentially going to find just more and more of these two lineages. It's not really going to add more value in terms of early identification or early prevention. What really needs to be done is to do genomic surveillance more and more in times when there are no cases across the country or across the world. And of course, that needs to have tools to do rapid genomic surveillance. And this is one of the approaches that we sort of developed in the lab, which uses benchtop sequencers, not NovoSeqs or HiSeqs, but small benchtop sequencers of the grade of MySeq and below, which can now sequence small number of samples, 48 samples, and give a turnaround of 24 hours. And there are hundreds of such machines across the country. And the idea is to sort of propagate this protocol so that individuals can, or labs can actually do faster genomic surveillance of local outbreaks and stop in their tracks and not really wait for weeks or months before the national surveillance kicks in. And this protocol is publicly available, and I'm sure if anybody would have a sequencer, you could suggest this protocol to them. And we have now extensively used this protocol in outbreaks in India and also across the world in different parts in regions adjoining India with the WHO Southeast Asian Regional Office, for example, surveillance in Nepal and Maldives, where we have effectively used this rapid protocol to sort of identify outbreaks, to do rapid surveillance of outbreaks, and to give much faster turnaround for the results for genomic surveillance. I think I'll stop there and take questions. But before that, let me acknowledge a lot of people who made this possible, including fantastic collaborators, Sridhar for most of the NGS and the lab activities, fantastic team of young students from Sridhar's as well as my lab, collaborators without whom this would not have been possible from across the country, and of course, multiple different consortia we're part of, including the INSACOG consortium, and funding from multiple different sources, right from the CSIR, the state governments, DBT, and of course, the WHO Southeast Asian office. Thank you very much. For students who are interested, there are of course multiple resources which have been created and publicly available, which you could have a look at. Thank you very much.
Thanks, Vinod, for that amazing talk. And I'm sure the students